still see people logging on. So we'll give people just a minute to get logged on. Hibret, my usual disclaimer is that nothing presented here should be shared outside of the group without your permission. I think that Tyler, who's helping me with my tech stuff, asked you if you're willing to have this posted on our site as a video afterwards, and I think you said yes. So I think you're okay with this being publicly disseminated. Is that correct?
Yes.
Excellent. So this will be available after the fact for people that want to refer to it on our new and growing AGP website, which maybe I'll send out the link again to follow the meeting. We're still working on updating the mouse pathology courses, and a number of you have volunteered to update individual lessons. I'm going to be asking again for that because we're going to start that process in earnest here in the next month, try and get it geared up to go live at the beginning of the new year, using, we hope, sort of newer technology that's a little bit simpler than the old Moodle platform that we used. There's going to be an opportunity for some of you to provide voiceovers for the PowerPoint style slides and or whole slide images. But there's also an opportunity for you just to provide the content updates, and we'll work on the voiceovers here. So stay tuned for all of that, and we'll be in touch.
Is there any timeline for that, Sandy?
Yeah, I'm gonna try and get. You know, it can be a dynamic process. So I think a lot of the content is very good and could go up. Some of it is dated in an okay way, and some of it is dated in a not so okay way. So we want to fix the stuff that's the most important first, and start to put it live at the beginning of the new year. All right, looks like Corey was able to log on, so I think we can start. I'll turn it over to you.
Okay.
A man who needs no introduction, although he moved from the NIH to Covance.
Yes.
I don't know if you want to tell us that story. That probably take too long.
No.
But I turn it over to you.
All I say is government is interesting, and I will move on. So, so today I'll be talking about male infertility and in GMs, and mostly I would like to talk about some of the observations we noted when it comes to morphology, and some of them really reveal the underlying mechanism or molecular pathogenesis. So I thought it's really cool to share that, so that we basically there is infertility at the background, but it comes in different flavors, and I thought some of the pathologies are really cool, and it also shows us how this could be applied to either to contraceptives or treatment of infertility, or for whatever reason that may be. But I thought this is really nice to talk about. So quick disclaimer, I think this work was done, most of this analysis was done when I was at the Hospital for Sick Children and the Toronto Center for Phenogenomics. When I was working with Colin, we are still working on this, and we continue to work on that. So this is where it happens. The acknowledgement, again, as I mentioned, it was done at TCP and when I was at Sick Kids Research Institute. I would like to take really time to thank Colin, who's also today attending this talk. So Colin, this is your own party. So I really appreciate for the opportunity that working with Colin and the great many things we did together, and we still continue to collaborate. So I appreciate that. And any question you have on this, you can also ask Colin. So Colin, if there is any difficult question, it is for you. And also I would like to thank Susan, who was a colleague pathologist there with me, and the director of pathology there, great colleague, I appreciate that. And Lily, a histotechnologist and lab manager, I cannot say enough about her competence and making this work possible. And most of the works today I will be presenting are also a collaboration with the Wellcome Trust, with the Sanger Institute, and Jackie White now at I think at Jackson has been very instrumental in this collaboration. All right. So male infertility is a global health problem. About 15% of humans couples face this. And nearly half of these cases are attributed to males, and the underlying cause and molecular mechanisms are not known. So most of these are really classified as idiopathic infertility. And genetic abnormalities are believed to underlie a large proportion of this, testicular failure and failure of spermatogenesis. But this is not surprising, as you know that almost thousands of genes are involved in spermatogenesis, so there is no surprise there, especially if you compare it to ovogenesis. But if we understand the function of these genes, I think it may shed light on some of this idiopathic infertility, from diagnosis, treatment, potential targets for male contraception. So it's important to understand that. And given the complex cellular interactions, as you know, with spermatogenesis, mouse models provide an attractive alternative to us to study this, because also mouse spermatogenesis is very comparable to human spermatogenesis. There are essential differences, but still the process is almost the same. And it's estimated that almost 50% of genes in mouse genome may be involved in testis development. It's really remarkable. I don't know how that was determined, but that is really remarkable if it is any close to the real thing. So about 4,000 genes may be associated with human male infertility. So this is really incredible. So several hundreds of fertility essential genes have been modeled in mice, which has led to the identification of infertility genes spanning all aspects of spermatogenesis, from spermatogonia development, proliferation, sperm maturation, capacitation. And so it can happen at any level, from migration of spermatogonia to finally fertilization. So it is really quite broad. So the IMPC basically, as you can see, this international effort, the idea is to generate a systematic encyclopedia of mammalian gene function by providing phenotype information on a knockout mouse model line for every protein coding gene in the mouse genome. So most of the ones I'm talking today are really driven from this effort, and the one in arrow is the Toronto Center for Regional Genomics. Most of this work was done. So the phenotyping pipeline, I'm not going to spend much time on this one, is really a battery of tests where these mice are exposed from 9 weeks of age to 16 weeks of age, and then different tests to pick phenotypes, or actually abnormalities from in this mice. And one thing unique here highlighted for histopathology is not a mandatory test for most of the centers. So TCP or Toronto is probably one of the centers which does pathology, and really you can see why it's really important to do pathology for many reasons that it's not time to talk about, but for reasons most of the centers do not really do pathology. The fertility testing is really what is probably important for this talk, and then I will mention a little bit about that in fertility. So the fertility testing, the protocol is done when mice are between 8 to 14 weeks of age, and the test duration, that means the mating period, is between 4 to 6 weeks. This is it. This is during this age limit and during this specific time that they will be met, and we try to determine whether they are infertile, subfertile, or fertile. So the primary screen is that there is a minimum 1 male homozygous knockout, and then a female homozygous knockout are made. If there is successful mating, then the strain is designated as fertile. The other thing is that then if minimum 2 male homozygous and female homozygous fail, then the strain is designated to be infertile. And then if the line is designated infertile, then we have to decide whether it is a male infertility or a female infertility. For that, there will be a secondary screen. For the secondary screen, there will be minimum 2 males homozygous. They will be crossed with a wild type female, And then if there is any failed mating, then this is designated as male-only infertility. And the opposite, minimum 2 males, 2 females, homozygous, will be crossed with male wild type, and failed mating will designate as female-only infertility. If this, in both of situations, if male-only or female-only failed matings happen, then the strain is designated as male and female infertility. So this is just to showcase you, like, to really to demonstrate, what the infertility phenotyping is, and then how these mice I'm going to show you are called either infertile or subfertile or whatever that designation was. So a little bit of something to think about for us is that, infertility of mice and humans is a little bit different. Infertility in the mouse, at least by IMPC designation, is inability of 8 to 14 weeks old mice, the ones I just described, or call them breeding pairs, to have pups in 4 up to 6 weeks of mating. Infertility in humans, at least by the World Health Organization definition, is that the inability of sexually active, non-contracepting couples to achieve pregnancy in 1 year. So think about that difference. That means, what does it mean really when we translate male mouse infertility to human infertility, and when we try to understand these genes, and what does it mean when we find different kind of lesions, multifocal lesions, all those kind of things that I will talk about. So just for you, some thoughts. So one thing we noticed, at least, is that infertility in knockout mice, there is a strong male bias. And this is interesting because if you think about it in human infertility, it is, at least, the predominant literature says it's 50-50. And, really we can spend an hour discussing whether the validity of that, considering what does it mean to be for a male to be fertile versus for women to be fertile. Socioeconomics in most parts of the world, that is where I come from, women undergo a lot of strenuous life, hard work. I don't know how that would affect their cyclicity, menstruation, and all those kind of things. And then, is it really genetic or environmental? Really, that is to be debated, because most of this data comes from, in the Western world, where probably that may be less significant, but still, I think women will face a lot of environmental factors that may affect their infertility. So I don't know whether this is genetic causation or environmental, but that's, something we can probably discuss down the road. So if we take infertility in knockout mice, there is again the strong male bias here. This is the current IMPC data showing about almost 6% of male knockouts have male infertility, whereas about 3% or 2.83% have a female infertility by that designation I told you earlier. So there is still the preponderance of males being infertile. And again, that makes sense considering how much energy, how many genes are involved in spermatogenesis versus ovogenesis. So this publication was in 2013, where Jackie published in Cell showing, their phenotyping result. You can see that, most of the infertility, they have 5.2% of the 307 lines they analyzed were infertile, and there were only 1 female which had only female infertility. 3 of them have both male and female, and 12 of them have only male infertility. The same thing probably, we did a collaboration with Sanger, with Jackie's Institute when she was there. And then we analyzed 275 mouse lines from TCP, 65 of them, and from Sanger, 210 lines we analyzed. We analyzed 2 males, 2 females, from 29 to 42 tissues, comprehensive histopathology, and this is what we found. So we had, from the 275 mice, total infertile lines were 14 of them here, as you can see that. Male infertility was found in 14 of these. That means all the infertile lines had a male infertility. There is 1 line which has female infertility and also male infertility. So as you can see, but very interesting is that if you take gonadal pathology, either pathology of the testis or the ovary, 27 of the lines, or about 10% of them, had testicular or ovarian change, but that's not accompanied by infertility, something we'll talk about. For the infertile lines, again, 13 of the 14 infertile lines had a gonadal pathology. For the males, 13 out of 14 had gonadal pathology. There was one line which we could not find morphological correlate to infertility. That may be functional, as I mentioned earlier. For the female, there was one, and it had also a pathology. So not to spend so much on this, one thing you notice is that only 13 of 27 lines with gonadal pathology had a record of infertility. That means there were 14 of them which had pathology, but the pathology was not severe enough to cause infertility. And that one is very important discussion that I will bring later, what it means. So the female infertility line, this was MCPH1, this is microcephaline. This is known, I think, associated in humans with having small head or small brain. But in mice here, because this gene is important for DNA repair, what we noticed is that, if you see the ovary and the uterus here from the wild type compared to the mutant, you can clearly see that, the ovary is basically hypoplastic. It is just vestigial tissue there. The other tissues are also hypoplastic, and this mouse is infertile. This is the only line we had infertility, female infertility, at least from the 275 that we analyzed. Now, before I move into the male reproductive, issues that might really mean the talk of pathology, I would like to say something about spermatogenesis. There is a saying, an African saying, that if you want to go fast, go alone, and if you want to go far, go together. I think for the sperm, I think it has its own definition. If you wish to travel far and fast, travel light. So you can see that, from this spermatogonia, the spermatocyte, it is consecutively what it is trying to do is that basically a DNA propelling rocket. That is what it is trying to do. It packs itself with mitochondria, jettison all kind of organelles later as they will be like, residual bodies, and then literally try to get this one sperm from all the way from the vaginal entry into the oviduct. So you can see that it has to travel light. So that is a little bit of difference between the African proverb versus what sperm thinks about travel. So here you can see that, this is a cross-section of the, seminiferous tubule. You can see the different cells from the spermatogonia, cervical cells. Progressively, you can see from spermatogonia, spermatocytes, spermatids, elongated spermatids, and sperm. So this is important. Why I'm showing you here is that depending on what kind of gene that is affected, the effect could be at the spermatogonia, at the spermatid level, or even at the elongating spermatids. And I will show you genes that affect these specific processes. And then when you see those under the microscope, at least even if you don't know the gene, you can tell that probably this is a process that's affected. And it's important sometimes even from drug development point of view, because sometimes we don't know in safety space that, what kind of test article effect we are looking at, but at least knowing what kind of pathology we see is important. So keep in mind that these processes are important. So today, I mean, really, not to make it, a lighter topic, is infertility is very, it is a devastating health and societal issue. But, if we have, I just discussed different flavors. For example, whenever under the microscope, what we see is that either seminiferous atrophy, degeneration, the garden variety we sometimes see failure of meiosis, failure of spermiogenesis and spermiation, germ cell exfoliation. It could be part of degeneration, but sometimes it comes as a specific effect, and it has to be documented as such. Multinucleated germ cells, we see them as part of degeneration, but sometimes they become a prominent and sometimes even the only features in degeneration. So I thought I picked those and tried to discuss, was relevant to the mechanisms that are affected. So this is really an excerpt from the book chapter that we are writing on this. So the genes associated with tubular atrophy or hypoplasia, this one in humans, I think sometimes called Sertoli cell only phenotype. I think it's also now used in mouse and rodent literature. So most of these genes involved in premature premeiotic germ cell migration, development, proliferation, differentiation. So this is at early stage. So if you see some of the examples we have here, if you see they affect, at an early stage of like primordial germ migration or proliferation. So those ones, they manifest themselves as tubular hypoplasia, atrophy. I don't want to distinguish those because it is a little bit murky there, but most of them, they manifest themselves as only Sertoli cells. And that's what what we see. Here is an example, for example. This is a nanos gene, and it's RNA binding protein essential for fetal male germ cell development. So you can see that it is important for male germ development at the fetal stage. So you can see that there is no germ cell here. All you see is that Sertoli cells, you see them lining up here. You can identify them by their prominent nucleolus. So then the whole thing is basically this scaffold of cytoplasm of Sertoli cells. So when you see this, when there are even no germ cells, it tells you that this is at an early stage of sperm development that the target has been. So this is tubular atrophy or hypoplasia. You can now, because now we know we can say hypoplasia, but we don't know whether it is postnatal or prenatal. So I would not go into those distinctions. I put some question mark here for you. These are, the Leydig cells, whether they are hyperplastic or not. That's something we can discuss later. Now, but sometimes what we see is that we still see tubular atrophy and hypoplasia. And then, of course, here is the epididymis being blank, and here is, you can see this completely atrophic or hypoplastic testis, and you can see the seminiferous tubule, the same, almost it looks like the same. But there is some difference between this and the previous one I showed you, and I will show you why. This is MCPH1. This is the one I showed you that there was hypoplasia in the female. This is the only gene we found, both male and female infertility. And this is DNA damage checkpoint and mitotic entry. Notice that this is mitotic entry. That means at least the germ cells are here and moving forward into spermatocyte and everything. So they are stopped at the spermatogonia level. So if you notice this is not a Sertoli cell only phenotype, because if we get closer, you can see that there are really germ cells here. See them there, see them there. And then there are Sertoli cells with their prominent nucleolus. So this one, I wouldn't dare to call it Sertoli cell only, but there are germ cells. And the previous one I showed you, since it is affecting even primordial germ cell migration, it was absolutely a Sertoli cell phenotype. So depending on where the gene is, you can see that even this apparently the same kind of morphology can be distinguished. And it's important to document those. So as I mentioned earlier, there was a little bit of Leydig cell hyperplasia in the previous one, but this one is really quite prominent. And here too, it is the same thing. This is a Sertoli cell phenotype. This is a gene, a transcriptional repressor. So it is really early in the spermatogonial development. So there is nothing here. All you see is that really clusters of Sertoli cells, either lining where they are basically clustering within the lumen. This kind of thing has been recently been described in black 6 mice. It can sometimes happen, these Sertoli cell clusters. Anyway, you can see that there are hyperplasia of the, so is this real or not is really sometimes the question we ask, because with the collapse of the seminiferous tubule, Leydig cells could be prominent. So there is a question whether this is a relative prominence of Sertoli cells, or is it really inherently hyperplastic? And in this case, it looks like it, that really they are quite prominent. And something, at least my own personal observation is that when it is Sertoli cell phenotype, we see them more prominent. It looks like if it is a hypoplastic stage at an early stage, I think Sertoli cells take over. But if it is at a later stage, like postpartum, postnatal, and if it's atrophy, they will still be prominent, but they will not be as prominent. So there may be some distinction, but if I cannot put my money on that. But I think I noticed this kind of trend where we see this hyperplasia in mostly with Sertoli cell phenotype. It looks like they started earlier. Anyway, so remember that sometimes what I showed you earlier is basically when everything is wiped out, you have nothing left. But you can also see this degeneration and necrosis as it goes on. And then it doesn't have to be even diffuse. If you notice here, it is diffuse. Here, it was diffuse. But sometimes you don't find it as diffuse. You can see that there are good tubules, at least trying to do their best. And then you have these multifocal lesions. I will discuss that. And then here, active degeneration of either spermatocytes or spermatids is happening. I cannot tell what cells are, but it looks like, this either late spermatocytes or spermatids are being targeted. And this lesion is not associated with absolute aspermia or lack of sperm, because there is a little bit of sperm here, and this is hypospermia. And some of these mice, probably most of these mice, are really fertile, and this mice is fertile. We'll discuss the relevance of that. But as I mentioned to you, not always it is a diffuse thing, and we find it as multifocal lesion. Here, for example, in this one, it is a transfer RNA synthesis affecting gene. Here you can see that there are multifocal, Sertoli cell phenotypes, and then here you have almost normal one. And most of the mice, I do not know any mice which was not, which was infertile. At least there may be reduced fertility, but most of them are at least, fertile for this one. So it can come as a diffuse or multifocal, and there is important distinction between the two. And here also you can see how, there is decreased hypospermia, and then maybe that's why it's called reduced fertility. Something which I would say is that probably most of you know this, but if we have younger colleagues here, we sometimes this area, this is the area of the, the straight seminiferous tubules, where the seminiferous tubules become straight and just dumping into the red testis. The lines, they line up with this Sertoli cell lined tubules, and they may look like degeneration. So stay away from this area where there is a capsule, because those areas look like degeneration or atrophy, but these are normal structures. They should be avoided. At least, don't, score your, necrosis or degeneration in that area. This is normal structure. So what does this multifocal degeneration atrophy, or what is its significance? One thing, it may be associated with hypospermia, as I showed you, and then there may be, infertility or subfertility, but often these are not associated with infertility or subfertility. Remember the 13 out of 27, the one I told you that we found pathology, but there is no infertility, those ones. So they are not really associated with infertility. Now, one thing I would like to say is that rodents have massive reserve capacity for spermatogenesis. So hence, focal or segmental lesions may not have notable consequence on fertility. So this is important. And often, quoted observation is that rats can remain fertile. They can produce offspring with less than 2% of their normal sperm numbers. So this is very interesting. Like when we do sperm analysis in some of these DART assays, I came across, I have seen worse. Recently, I had a study where there was this rat, which, I think everybody claimed that they were fertile. There were pups from this one, and I could not find a single germ in its testis, sperm. And then it has a pup. So, I just said, this is really my pup. So you can think of, a lot of possibilities. Is there parthenogenesis, murine, immaculate conception, or, there was a milk mouse, like, the milkman, who knows what. But it is really interesting to me that, that rat was, I call it the curious case of rat 201, jokingly, because it was fertile. So what does it mean? Will it happen in humans? And that is something we have to think about. What does that mean with relevance to rodents versus men? What does it mean? So quickly, what I would say is that, remember, I told you that, fertility testing happens it is a specific window of period. And most of these GMs, wherever, I don't know, in your institute or something else, they are terminated at a relatively young age. And we never do longitudinal or temporal analysis in this one. So we do not know, later they become fertile, or they are fertile now and they become infertile. We do not know. So we do this cross-sectional study anyway. How about in humans? For humans, there is potential for age-associated progression. We have long reproductive lifespan. So what happens if there is a genotype, for example, a gene which causes focal degenerative changes? Will it progress, considering the lifespan? So that is something, when we translate to humans, we have to think about. And the other thing is that humans have inherently relatively low reserve and efficiency for spermatogenesis. So they are not like rodents. So we have to think about that. Those genes which we think are multifocal or focal with fertility could be consequential in humans. Something to think about when we translate that. So the next ones I will talk to you about is that failure of meiosis. So I told you like earlier, when spermatogonia are affected, what you get is that these Sertoli cell phenotypes or hypoplasia or atrophy of completely. But when meiosis is blocked, that means between the from spermatocyte to forming spermatocyte, this is reduction division. Sometimes some genes affect this specifically at this stage, and then I'll show you what. Some of the genes, at least a human phenotypes for the same genes versus, for example, for the mouse models. Here, for example, ATM text, those are known to cause meiosis arrest. And there are comparable phenotypes between humans and mice. And some of the examples I will show you are here. For example, when you have a defect in failure or failure in meiosis, what you see is that you see a lot of spermatocytes, you see them here, they are ready, but then they don't progress beyond that. So clearly you can see that, there is a spermatogonia, everything goes on, and then there is spermatocyte and stops. So when you see this under the microscope, it should alert you to a possibility that there is a meiosis arrest, for example. And visually, this one leads to maturation depletion. At least, in books, most of the time they use maturation depletion. That means any of the germ cell lines after whatever line is stopped will not be there. That means there is no here spermatids, there is no elongating spermatids, there is no sperm, so like that. So they will be, the consequence steps are affected. And here is the epididymis with basically no. And for this gene, SPO1, this initiates meiotic recombination by generating DNA double-strand breaks. So it's very important for meiotic synapses happening. So without that, it doesn't progress beyond this. How about failure of spermiogenesis? Now, next step. Now you got to spermatid, and then something stops. From here, it is a differentiation step to become that spermatozoa. And here, there are some genes in humans as well as in mouse models that do that. And then what happens is that it could be sperm chromatin condensation, it could be transcriptional regulation specifically to that process, especially like acrosome maturation and those. So I will give you some examples of that. Here this is a defect of failure or spermiogenesis. This is a maturation depletion again, but because of spermiogenesis defect. So you can see now, compared to the previous one, I will take you back one time. Here, you have here only spermatocytes. Now you move on to this one, what you have is that you move from spermatocytes, which are here, the darker ones, you can see these packeting ones, and then now you have the spermatids. But what you don't have is that there is no elongated spermatids. There is no maturing sperm. So this is, for example, a cyclotogranin. It is a chemotaxis function. We are not sure really how that happens, and then you have that arrest at spermatid. So this is also have to be documented. So it tells you that probably this is a differentiation affecting gene. And you can compare it to the wild type. This is what you should be expecting, but then everything stops at this level. All right. And then sometimes this can happen, and we have, for example, in this one, in GNPDA1 long gene, this plays a role in acrosome reaction. Remember the acrosome, I told you this is basically here. These are the few organelles it has for the nucleus to propel, and here is the mitochondria. Sometimes what happens is that the defect could be in elongated spermatids. So everything is coming well. There are spermatids, and then once the elongated spermatids they just die. And this is, I have never seen any other line with this phenotype. And there is a toxicology case with this reported previously, but I have never seen it in a mouse GM where elongated spermatids all of a sudden die out. So this again affects the spermiogenesis because it is the second step that's affected. So you can see that, if you see this in your line, you would be aware, oh, maybe this is an elongation thing. This is a specific gene expressed in that. And indeed, for this one, it is acrosome. So it is the acrosome that is supposed to be working out when it's mature and it's not happening. So that's a cool example of that. And then, of course, there is no sperm in the epididymis. But something which I would like to emphasize is that this should not be confused with the atypical large mouse residual bodies. Mouse residual bodies, unlike rats or other animals, are really prominent. So let's not confuse those with elongated spermatid generation. You can see these ones versus these ones. So just to be aware. And then this is another cool one where this is a failure of spermiation. Now, if we say that once the spermatozoa is made, that means it is elongated, it has to, it forms this flagella and it has to be released. But sometimes what happens is that it stops from releasing. Some of them actually are embedded here deep down in the to the basement. You can see some of them here. And there are two, so you can see that there are some elongated ones here, and you also have some completely elongated spermatozoa. This is no, because you can't have two elongated versions of germ cells. This is a maturation issue. So this is a failure of spermiation. It doesn't, they don't release. They are trapped back and taken, probably even phagocytized by Sertoli cells. And this is what you see in the epididymis. There is nothing. So sometimes this could be tricky. It could look like normal. So you have to pay attention and try to see how many elongated, germ cells I have, how many non-elongated, and then try to see where they are. And you'll find them here embedded in the deep lines. And that is really a very important one. That's what they call sperm retention. Sometimes it is retained and phagocytized. All right. Now, so let me talk about this gene. This gene is a centrosome protein 41, and it is important for flagellar development and motility and that kind of function. It is associated with actually ciliopathy. So some ciliopathy genes, they are associated with sperm retention or motility issues, both ways they could be. So here, the laxity expression, you can show strong gene expression in the testis, but it's also expressed in the, in the choroid plexus, lining the choroid plexus, the ventricles. And this mice line had also hydrocephalus. So this is probably, a PCD, a primary ciliary dyskinesis situation where there is lack of motility, but some of them, they may have retention of sperm. So this is really a cool example. Now, this is one of my favorite ones. Sometimes degeneration comes in different forms. And one of the type of degeneration, when you characterize, you can say degeneration, and then there is exfoliation of cells. It can happen because these germ cells, when they are not supported by Sertoli cells or they have problems, they just physically shed because they have cell-cell adhesion between them with the Sertoli cells because they grow as a pack. And then when that is disrupted, they could be shed. It can happen under any toxic or genetic lesions. But when you see it as a very prominent feature, you have to document that. And why it is a primary kind of exfoliation is that when you look at the epididymis, you see a lot of these round spermatids basically packing the epididymis. So be careful because you may find this kind of thing using tissue processing. So that's why sometimes it is we use Davidson or that, but this is done with formalin fixation. It can happen because of fixation, but this is not. You can see that these germ cells are dissociated from their siblings and basically exfoliating, leaving too early. This is very important. And the gene is a cell adhesion molecule 1, very important link between Sertoli cells and germ cells. So they can live. How you can differentiate it from artifactual or the lesion or caused by artifact is that look for the epididymis. In artifactual situations, you will not find these germ cells in the epididymis, I mean, left around spermatids. But this is very cool here. I'll show you that. This is, I call it, the lesson on contraceptives from the armadillo. This is, the hairless armadillo. This drawing was done by my daughter some years ago. I paid dearly for that, really. But the issue was that I cannot use any image when I was at Covans. We cannot use, online images because of, all the issues of copyright. So I asked her to draw this for me, and that's what she did. So the point is that this armadillo has this very interesting seasonal contraceptive device mechanism. Basically, when the season is no more breeding, it downregulates all these cell adhesion molecules and sheds off all the germ cells. And basically, that is how it controls, unnecessarily waste, it is resources on spermatogenesis when it doesn't need it. So the same mechanism, one of the genes, which actually it is CDM1, this gene I talked to you about, is one of the ones which is downregulated. So it's really cool how, biology works across the mammalian genome. Well, the next one I'm going to talk about is that multinucleated germ cells. These are symplasts. They call them symplasts because, remember, germ cells develop as syncytia. They grow together as a nest and nurtured by the Sertoli cells. So occasionally, we can see these multinucleated germ cells, commonly in young ones. If you see, in mice which are about 4 months or younger, you can see 1 or 2 multinucleated germ cells, which we call symplasts. That's fine. And they are also considered, if you see them more, they are considered at least slow degenerative process. At least, Creasy and colleagues, they think that it must be some kind of slow degenerative process that leads to the formation. But we notice that this feature may be dominant and sometimes even the only feature in some infertile GMs. And I will show you some cases where these symplasts are the prominent feature. So this is normal. You can find some of them like this, probably remember, the first wave of spermatogenesis is faulty. The testis is setting up a shop, germ cells are coming, they don't know how to multiply, they don't know how to control their DNA repair. It is faulty, it's just like kids. So this, degenerative, symplasts can form. That's fine. But when you see them a little bit more, we see them, for example, with this typical degeneration. When there is exfoliation like this, for example, for the cyclin T2 gene, you see that this exfoliation, you see them, the same thing like I showed you, exfoliation. You can see like a couple of like this. They are not as many, but you can find a couple of these, multinucleated symplasts. Basically, they cannot separate from each other. They are degenerating and they are packed together. But then there are some lines like this one, for example. This is a PEX3, peroxisome biogenesis factor, very important for degradation and oxidation of fat. So I'm not sure whether it has anything to do with membrane function, but there's no other phenotype in this mouse. But you can see that this is basically all over the place. You don't see even anything per se degeneration. All you see is that these multinucleated germ cells, some of them look apoptotic or necrotic, but this is what you see. And also, if you see the epididymis, you can see that ghosts of those, literally. And then we try to do some tunnel assay to see, if this is really, there is apoptosis going on. Really, it is rarely that you find those apoptotic multinucleated symplasts. Most of them are at least viable, but degenerate. And this is a feature for this one. And after I looked at this one recently, we have not published this one recently. Actually, what happens is that we got this mouse at least at 6 months old. This is when they were, when they were 16 weeks, and then now we got them a little bit older. The same thing. This process happens even at the marked rate. You can see these multinucleated germ cells. So this seems to be a specific feature of X3 gene, or maybe peroxisome biogenesis factors. And a group from Germany recently published that they knocked out PEX3 specifically in the germ cells and found exactly the same thing that we were observing with PEX3. So it is really interesting how this gene, so we suspect that probably these PEX genes, because of their specific function and probably in membrane degradation or fat oxidation, their absence may lead to coalescence or not breaking of those membrane bonds and maybe form syncytia. We are not sure, but this is a feature we see in some of these mutant lines. And the most interesting thing is that there was a study in Drosophila when they knocked out these PEX genes, and guess what? They found exactly the same thing. PEX10, PEX3, they formed this multinucleated cancer. So across different mouse strains with PEX, different PEX genes, even in Drosophila, cause this multinucleated symplast. So we suspect that, we know that this could be a garden variety of degeneration. We admit that. but there may be some specific pathways which may lead to degeneration with this specific symplastis being the predominant feature. So it's always good to document those when you see them to be the most prominent feature. If you see one or two of them, it's really not worth it because we see them as part of degeneration. So in summary, really, infertility is more frequent in the male mutant mice. I think, I showed you the data. Testicular pathology is present in most cases of male infertility. And sometimes testicular pathology occurs in absence of infertility or fertility. Like 13 of the 275 mouse lines, almost 5% of them had testicular pathology of some sort, but nothing really, there was no fertility issue. So it is important, think about that, what does it mean with its progress, considering we do only specific age and then for a specific period with this progress, what does it mean translatability to humans? Those things we have to at least be cognizant of when we make those model validation. So as I showed you that testicular pathology is varied and mostly reveal underlying genetic lesion or molecular pathogenesis. So whenever we are looking at, this testis from mutant mice, it would be nice to do stage-aware analysis, trying to see where in the line, at least, try to see what lines are not and what are not represented. So if we say which germ cells are not there or not, and do a stage-aware assessment, we know at least what areas are being targeted. So it's something to think about. So again, of mice and men, as I said, infertility affects almost up to 7% of all male or men, especially when it comes to that. And male mouse in the current and previous study, again, predominance of that pathology without infertility is there, as I mentioned, and I wonder about delayed onset about that. And the question you may ask is that why male bias? Again, 50% of the genes in the mouse genome may be involved in testis development, about 4,000 genes associated with human male infertility. So you can tell that it's really a very risky business to have a safe spermatogenesis because anything can go wrong kind of situation. And the highest frequency of gene expression in the mouse testis, there was a study we did with, I think, UC Davis and also Sanger about gene expression in this mouse lines, and 42% of mouse genes are expressed in the testis compared to 23% in the ovary. Just literally half of. So it looks like, testis or, spermatogenesis needs more genes. And about 8% of the mutants that were analyzed had gene expression only in the testis, and it was not seen in the ovary, not staining exclusively in the ovaries, never observed. So it looks like the testis or spermatogenesis is highly dependent on many genes, and maybe that's why we see more of those issues. So before I close my talk, for this chapter we are writing, we have enough material for infertility, but we lack material for the tumor section. So if you are willing to share any GEM testicular tumors that you have, and you can share that we can use in that chapter, we'd really appreciate, and we will duly acknowledge in the text. So we're looking for that, because most of these analyses we do are in young animals, and we don't get them to a level where they develop tumors. And then finally, I would like to say thank you. And this is an image of an ovum. As you can see, it really looks like an anaconda or something. These are 3 ova from the same ovulation, but I think the male was infertile, so I called it waiting in vain. So this can happen. So with that, I finish, and thank you for your patience. I might have gone a little bit over time because I'm so passionate about this, and as you can see. So
no,
any questions I have?
Good job. That was a tour de force.
Oh, I appreciate it. Okay, and thank you. So if you have any question, please let me know, and I will try to answer any difficult question. Please ask Colin. Colin, you are not away yet, right? So Colin, you will answer the difficult ones. The easy ones I will. Thank you.
That was really good. I learned a lot. I've seen testicular atrophy of various kinds under the microscope, but never been very good at classifying it. So I'm well armed now. I'm gonna have to go back through the video to refresh my memory. But I have one question, which is,
Sure.
in the pipeline that you described at the very beginning, there's the functional test for male infertility, which is great.
Yes.
And maybe you said this and I missed it, but how often do you have functional male infertility, but you don't see anything at all that you can find wrong with the testes or the epididymis or any of that?
That yeah, I think probably I briefly mentioned. I know I glossed over that. I know you would ask. I think. Okay, here we go. So if you see here, let me just change it here. You see that, for example, one of these lines, this male infertility. If you see that there were 40 lines.
Yeah.
There is one, no microscopic abnormality. And this one, we couldn't find any. And then I think we suspect that there wasn't, I mean, the testis, the epididymis was full of sperm. There was nothing there. But I think probably it may be this functional abnormalities that I think in humans, they describe them as MMAF or something like that, this motility and morphology abnormality features or something. There is a human, I think, probably.
Yeah, you know, it's not my area really, but
Yeah.
I know that in humans, we will do functional motility of the sperm
Yes.
in vitro
Yeah.
as one of the assays. And I don't know
Yeah.
exactly how the assay is done, but.
I don't think we have that kind of data for this one. Colin can comment, but I don't know if we have the semen data from this mice, but it would be nice to see if they have any abnormality for some of the rare ones. But almost, you know, most of them we find a pathology, and sometimes also looking really hard because I always suggest that, you know, start from the epididymis instead of the testis, honestly, because the epididymis is what tells you. Even for the one line, which there was, you know, sperm retention, you know, at first pass, you don't see there is any problem. But the epididymis tells you there is no sperm here. Then you go back, you see all this sperms retained or the lack of maturation. So for those subtle ones, I always suggest that, you know, start from the epididymis and you will find it. But at times we don't find anything, and then I think we attribute to those to, you know, functionality.
And I can say that the part of each of these strains, as they're created as part of the production process, is sperm cryopreservation, and depending on the center, there is variation. But I know at TCP, certainly there's an assessment of the sperm, because there's no point in freezing sperm that isn't going to do anything when you reanimate it. So it basically is well, I think it's the IVOS system that we're using, and it's concentration, motility, and progressive motility. I think are the three primary parameters, and for a pass or a fail. So that could certainly, yes. And then there's, you know, always remembering a potential completely extraneous like failure to copulate, of course. I mean, if there are other reasons that that they can't get the job done, then you know, those are other considerations as well. So I'm surprised actually there's not actually a few more of those, because you know, there are multiple of these lines with pleiotropic phenotypes, meaning, you know, a gene dysfunction affecting many systems. So they could be feeling very unlike mating for a whole variety of reasons, but the gonads histologically look good.
Other questions? I really appreciate you giving this talk. It was very educational for me. We may need to turn this into yet another module in our course, because I don't think we covered it very well the first time through. So so stand by, Hibret.
I will.
And with that, I'll let everybody go. I don't have anybody signed up for next month. we run into Thanksgiving, so we'll have to avoid Thanksgiving. We'll do it on I think it winds up being the fourth Thursday is Thanksgiving and the third Thursday is not. I can't remember. We'll do it on whichever one's not Thanksgiving. Unless you guys want to have a meeting on Thanksgiving, since nobody can go see their extended families anymore. So stay tuned, but I need a volunteer for that. And I think there's a lot of interesting stuff out there. The thing that I posted as a postscript on the invitation is that some of these immunotherapy and knockouts of specific components of the immune system will of course result in various kinds of autoimmune diseases. And so I'm curious about autoimmune disease in the kidney and collecting some of the experience on that. So stay tuned on that. And I may have a collaborator for you that needs an expert if you're interested. And I think Jerry said he'd be happy to look at those kidneys, but anybody else, I don't have the slides yet, but I'll have them soon. I can share them via the Spectrum website. The Spectrum website has moved. Those of you who have tried to log into it have maybe seen that. And if you run into trouble with that, it is going to demand a new password. We've got more stringent password requirements. Let me know if you have any problems and we'll try and fix them.
Sandy, would you post the website for the course material again?
I will do so.
And the second thing is that from the early 1990s, we have a number of cases that potentially have Leydig cell tumors in mice. However, we do not have any whole slide images from that era. So
you want me to dig around in the archive is what you're telling me.
Well, whether it's worth, let me search the collection first and see if we have any JPEGs of these tumors.
Okay.
And they may be adequate for what they're looking for.
And if we still have the slides, they may be findable.
Okay. I'll take a look and post you. Over and out.
Thanks, everybody. Talk to you soon. Be well.
Thank you. Thank you, Sandy. I appreciate it. And thank you, everybody, for attending.
Thank you, Hibret.
Thanks, Hibret.
Thank you so much. I appreciate this.